So yes, thank you very much for this opportunity to present the work that I've been exploring in my lab along with my students, and in particular looking at gene expression and gene regulation during human brain maturation. So firstly, why should we study gene expression in the brain? My lab is really interested in this field primarily to gain insights into how our brains grow and function. We're particularly interested in understanding the molecular mechanisms that are guiding brain maturation, and this process is occurring during childhood and adolescence. So in order to do this, we first need to define the gene expression profiles of each brain cell type, and then what we are really focusing on is exploring how these gene expression profiles change over the course of brain maturation from birth until adulthood. And in addition to just looking at gene expression, we're also interested in identifying the gene regulatory elements that are potentially controlling these gene expression dynamics. And importantly, this information is not only just useful to understand how our brains grow and work in a normal, healthy state, but this exploration is also hopefully going to provide potential targets for understanding the molecular mechanisms of brain disorders and disease. So much work has actually already been done to understand the molecular profile, specifically in the adult brain. The efforts of the Human Cell Atlas so far, along with also the Brain Initiative Cell Census Network, have resulted in a really amazing draft of the cell types of the adult brain. And this cell census has specifically focused on the primary motor cortex and the middle temporal gyres. So to make this single cell atlas, they combine gene expression information along with a host of epigenetic data indicated here in order to define each cell type in the brain. So we have this amazing cell atlas, or at least a beginning draft of one, but what we don't know is how these profiles are set up. So how do the profiles change over the course of brain maturation? So previous work has tackled this question already using bulk RNA-seq. And what other researchers have found is that the most drastic changes in gene expression in our brain seem to be occurring during this late fetal and early infancy stage. However, it's possible that there may be changes happening later on that are being masked by this bulk approach. So there could be more cell type specific changes that are happening that we cannot see when we use a bulk approach. So at the University of Cape Town, we actually now have an opportunity to start testing this hypothesis and profiling the pediatric brain at the level of a single cell rather than in bulk. We have, so through an amazing collaboration we put together with clinicians and neurosurgeons at UCT and Cape Town hospitals, we are putting together a biobank of ante-mortem brain tissue samples collected during surgery, mostly to treat epilepsy. And importantly, this biobank now consists of tissue from the local population, and it currently spans ages from about 10 months to 46 years. And so the main point of using this resource is to build a locally relevant single-cell data set, which is now allowing us to explore how cell type specific gene expression in the pediatric brain is changing in comparison to what we know is happening in adults. To do this, my lab is taking two major approaches to explore gene expression. Firstly, we're analyzing these biobanked patient tissue samples using single nucleus RNA-seq, focusing on the 10x Genomics Chromium platform. And this is allowing us to build single-cell atlases for our samples and conduct differential expression analysis between samples of different ages during the course of brain maturation. So in addition to the single nucleus approach, we're also analyzing intact tissue sections from the same patients, and we're using spatial transcriptomics to analyze these sections. So using the 10x Genomics Visium platform, we're hoping to gain insight into the spatial dynamics that is occurring between samples, both at the level of cell types and the level of specific genes. And so in this talk, I'm going to be showing you our preliminary data from using both of these approaches. So firstly, starting with our single nucleus RNA-seq data, We have generated 6 new data sets, which include 4 pediatric data sets, and they're all from the temporal lobe. In order to increase the sample size in our analysis, we've now integrated these data sets with previously published work also from the temporal lobe. So what you can see here is the integration of these data sets using the Seurat pipeline, and you can see a clear mixing of samples, mixing of the nuclei across most of the clusters. We've then been using a combination of automated and manual approaches to annotate these clusters as one of the major brain cell types, and we've done this using the expression of known marker genes. Additionally, we've also been using Seurat's label transfer approach to try and provide a higher resolution annotation of the cells at the level of cell subtypes. And so this involves using the curated labels from the brain cell census and transferring them onto our data set, as you can see over here in this UMAP, where the clusters are now broken down into cell subtypes. And reassuringly, we can show that these annotated cell types are expressing the expected markers. So as you can see here, these are the markers for the inhibitory neurons, excitatory neurons, and your non-neuronal cell types coming up in the appropriate cell types. So our ongoing work with this data set is now actually to try and run de novo marker analysis on our annotated cell clusters. And this is to determine if there are any markers that are indeed age-specific, or markers that are showing major or subtle changes in gene expression levels across the different age groups. The other important question we're exploring is whether there are any spatial dynamics in the location or the abundance of these different cell types that we've annotated. And to do this, we're using our Visium data. So here I'm showing you our data using the cell-to-location tool to transfer cell annotations from our single nucleus RNA-seq dataset onto a matching Visium dataset. And in particular, what you're seeing here is one of our 15-year-old samples. So these plots are cell abundance heat maps, where warmer colors are indicating a high estimated abundance of the indicated cell type in the given Visium spot. And importantly, this provides validation of our single nucleus and RNA-seq analysis, because it's showing that the cell abundances correspond with their known locations in the tissue. So for example, if you look at the layer 5 IT panel, you can see that the higher abundances are deeper in the tissue compared to the layer 2-3 IT neurons, which are found more peripherally. And the same is similarly, if you look at the glial cells, you can also see that oligodendrocytes are mostly located in the white matter region, whereas layer 1 astrocytes, in the layer 1, you can see an abundance of astrocytes as expected. So using this approach, we can also now start comparing cell type abundances and spatial patterns across samples of different ages. And that's what you're seeing here. You can see merged cell type abundance plots for the different cell types. So for excitatory neurons, inhibitory neurons, and non-neuronal cells in both our 31-year-old and 15-year-old Visium data sets. And already you may notice some potential differences. It looks like there may be a possible lower abundance of layer 2-3 IT neurons in a 15-year-old sample in comparison to the 31-year-old sample, as indicated by these blue spots. There also appears to be a high abundance of astrocytes at the border between the white matter and the gray matter in the younger sample, which is not present in the equivalent region in the adult. So to further investigate changes in cell type abundance, we're also using cell-to-location's later clustering tool to identify regions that may differ in cell composition with the aim of comparing clusters across samples of different ages. So on the left is an example of this cluster analysis for the 15-year-old Visium sample. And reassuringly, when we visualize these clusters in the context of the tissue section, you can see they're clearly recapitulating the layered cytoarchitecture of the cerebral cortex. So the next step for both our single nucleus RNA-seq and our Visium datasets is to explore gene expression profiles of these identified clusters. and to determine if the profiles vary with age. So we've already started doing this for our single nucleus RNA-seq data. We're testing the DE-seq2 pipeline for differential expression analysis. So for each cell type cluster, we've aggregated the counts across the nuclei for each sample to create pseudobulk data. And then we've also grouped our samples into 4 epochs, which represent the early and middle childhood, as well as early and late adulthood groups. After conducting differential expression analysis, we then are grouping the significantly differentially expressed genes into modules that show different expression, that show similar expression patterns over time. And so here what you're seeing are these modules specifically for the excitatory neurons. And what was very interesting to see is that we could see among these groups, there's a module of genes that is specifically increasing in expression over time. And when we looked at what genes were actually encapsulated in this module, we saw that it includes a gene known as NR3C2. So this gene encodes for a mineralocorticoid receptor. And interestingly, this has actually previously been associated with aging in both bulk tissue and also in laser-captured Purkinje cells. So we're reassuringly confirming previously published data with this analysis. This group also includes a cell adhesion molecule known as contactin 5, and this is known to stimulate neurite outgrowth, and also in the past has been shown that it may play a role in synapse formation in the auditory cortex. Interestingly, this gene has been suggested to decrease rather than increase in expression in rats over the course of brain maturation. So, however, this previous work was done in bulk, and so it's possible that when you look at a cell type specific profile, you see a different response, or what we're seeing here could actually be something that's specific to humans when compared to other mammals. And so we're continuing to conduct this sort of analysis across all the different clusters that we've identified for our single nucleus RNA-seq data. And ongoing work in our lab also includes continual optimization of our analysis pipelines to detect differential expression using the latest published tools. We're also generating more single nucleus RNA-seq and Visium data sets in order to increase our sample size and increase the power of our analysis. In addition, and there wasn't time to cover this in this talk, but we've also generated bulk attack-seq data sets for the majority of our biobank samples, and we're preparing now to also do single nucleus attack-seq in a subset of these samples, so we can start exploring the chromatin dynamics that are likely regulating the gene expression changes that are happening as the brain matures. So in summary, I've shown you how we're using a unique tissue resource, which has been available through collaboration with clinicians at UCT to describe cell type specific and spatial gene expression dynamics that are occurring during brain maturation. And my collaborators and I hope that this will form a really important locally relevant control data set for understanding how cell type specific gene expression during brain maturation may change in response to locally prevalent diseases, and particularly those that affect children, such as TB meningitis. So with that, just to end off, I would really like to thank the really excellent master students in my lab who have done all this work that I've shown you. And so these include Stephanie Fillmore, Rabimbo Mishi, and Christina Stain. I'd also like to acknowledge my long list of collaborators without whom I wouldn't be able to do any of this work, as well as my funders.
I'm going to talk about establishing single cell as a platform. So I will start with a little bit as a story, because I've done similarly when I was working in UK. And actually, I'm talking to you from UK, not in Gambia, because I'm on my travel and looking at what work's been done in my old lab. So I'm really giving you a timeline and a story from setting up one in the north and one in Gambia. Okay, next slide, please. Do I do the slide or do you do the slide? Okay. Yes, just a brief summary of what I'm going to talk about. Genomics in our region, the challenges, and I look at it more as an opportunity to do some work. The MRC genomics platform, that's set up as a cross-cutting service. Some timeline comparison between what I did in UK and what I'm trying to do in Gambia. And then really to end up with this project that we've had with CZ Bio on a single cell project that I will talk about later on in my talk. Next slide, please. A little timeline on where I've worked. I worked in the MRC in London for a few years, set up a facility there after doing some bioinformatics course. So I was setting up a facility from scratch. So there was nothing when they asked me to do it. And moving that facility to the creek, and then moving to the Gambia, and now having a strategic position to do that. Okay, so just again, this is for the people who are actually really in this kind of conversation, is that we know genomic sequencing is taking a huge part, especially really during the COVID pandemic. It shows that sequencing has a huge part to play in biochemical research. And there is a huge interest actually, again, with people trying to explore African data set. Really, the big issue is that most of the work that is done on African samples are done outside of Africa. So that we need to change. And I was listening to Alex talk about Black Lives Matters, equity in science. And so really trying to make sure that we could do most of the important work that we need to do. There's a capacity for us to do it in Africa. It's a huge thing because there is a huge benefit in terms of training people and where we go. So next slide, please. And there's challenges in terms of trying to do this in Africa. There is really a shortage of expertise, and the technology transfer is really quite poor. And infrastructure, as people like Gordon and I experience, it's really very, very difficult. Things that we take for granted in the West or in the North, it's really very, very difficult to actually access. And accessing not just the technology itself, but even once you actually do have the technology, in accessing reagent, accessing the infrastructure to get reagents to it is really very, very difficult. And that really puts us at a disadvantage, in fact, in terms of really applying for funds to be able to try to do seminal work in our region. So then we ended up actually going for the free launch, where people do send sample away because the collaborators are on the north and they would do it for free in bracket. Next slide, please. But actually, there is a huge what I would call benefit in doing work in Africa. It means that, and I've been really seeing the benefits of that during the COVID, and we could teach each other when the borders are closed and nobody could move from the West to Africa. We could do that. I mean, I've been moving around trying to teach people to do their own sequencing. So we could do that. There's a huge potential for growth. And we also do have a huge access to the different types of samples. We know that African genome is really quite complex. So it means that African data is really quite important to be able to achieve. So really there's a huge opportunities for us to do genomic research, single cell research in our region. Next slide, please. And this is a vision that the unit have. It's not mine, but I was collaborating and supporting them before I moved over. And 4 years ago, I decided to actually take the plunge. And we started with a really complex model where the genomics facility sits within a triangular, but the triangle was managed by different governance. We try to simplify it with 2 heads. So I've been made a head of the strategic genomic, and there's a new data science head that's just been employed. So we will work together to really get the platform to be in a strong position to be able to deliver to the scientists in the unit. Next slide, please. And really what I said again, the unit is set up really well, and it's been around for a few years. I've been here 4 years, but the platform that I've trying to establish, we have a huge biobank. Samples being collected and been in freezers for many years that we can access. that have got really good data sets that we'll be able to access when we want to use the biobank. We have the units always developing in terms of the facility and our infrastructure is really well established, so we don't have issues of electricity, water, and lab spaces. And we've set up, and while I set up, high performance computing so that we could do our own data. And we collaborate with northern partners like Climb to support us to make sure that we have really the infrastructure to be able to analyze our own data. And we also provide this service to people in our region. So all the places I've been to do capacity building, they have access and for free for now to our high performance computing. Next slide, please. And the platform work across is cross-cutting. There's a couple of themes in the unit, and the platform have projects that we've developed in the last 4 years, and we've been able to show that having the genomic facility is actually a win situation. So people are able to apply for grants, and we've had over 20 million pounds of grants that have the platform as one of the primary service that would for the particular project that we're doing. So that's across theme, which really is what the platform should be science driven. It's not people buying equipment and parking them and not being able to use it. Next slide, please. And this is I just wanted to compare a timeline of the 5 years I was starting up a facility. And the way it works is like it's an MRC facility. So you bid every year for equipment, provided you can, there is history that the equipment that you buy is going to be used and is science driven. I was lucky. I thought when I was there, I was really lucky that when I started with a GA, I was able to get various equipment. So firstly, you need to establish that you could run bulk before you start going down to the single cell pathway, to be able to do really set up the facility to be able to do sequencing, to be able to analyze data, all of those things before you can start going a little bit higher with single cell. So we started and we were able to get a couple of fluidigm that were really running very well. Going away from that, I've been away for 4 years. They've actually not using the fluidigm anymore. They've moved on to really the science cell one and other, what I would call nano single cell technology. I'm not in that bracket because we're talking about costly thing, but it's shifting to say that the facility was really successful and there are really hundreds of papers that have come out of the work that the facility established in Mill Hill. So go back to the next slide, please. And this is just showing the lab in Mill Hill. So next slide. And compared to where I am now, so really I came, as I said, in 2017, and we also able to, with the same format, you apply for a capital bid from MRC, as is an MRC site, and really we also been doing reasonably well. And this has to be with a caveat, we have a Promethean, so we can do slightly larger data sets, generate larger data set. We are not where from in the creek where they've got an NovaSeq and they could do on the Illumina platform. But really, for me, again, I am a huge advocate for the ONT platform. And it showed really well within the COVID setup how well the platform works in our region. So I'm really advocate because access to reagents is a lot easier. And also the startup money that you need to start up the platform is really quite easy. So we are now in a position where we could also do single cell. We have a fluidigm, bidding to get things like a Julie stage and microscope from the people I'm working with. So we're still using support from the north to be able to set up the facility. And we also getting a 10x for some of the work we're doing. And again, we have, as the Sesay grant, which I would allude it later, is that we're going to also use some of the modern platforms in single cell, like SQL, and Alex is one of the my PI, co-PI in that project. Next slide, please. And this is the lab in MRC. We are very, very fortunate. We got the space, we got equipment that is got projects that we could do. For the Prometheum, I've got a project that's already wants to run about 800 flow cells in the next few months. So it's science driven. It's not packing instruments that we do, and really well supported by the unit director to make sure that we progress really well in terms of providing a service for the scientists. Next slide, please. So this is just a couple of reference paper because my name covers the side. It's reference of a couple of papers that were published in the work that we did that I was really active with. And there's a lot more just as an example. Next slide, please. Next slide, please. Okay, so we have secured a grant just before COVID, and it's a part of the Global Pediatric Cell Atlas, and we're going to look at the nasal and oral mucosa, and really involves 7 sites. And Alex Shalek, who was presenting earlier, and one of the I will call the brains in single cell is part of this grant. And really what we want to do is, and with COVID and in terms of what we do, a couple of our collaborators have been working on the nasopharyngeal for looking at viral infections, other infections in small children and in adults in our setting. So this really would work really well. So it's a small pilot project, but really something that brings us in, that we're part of a network. We will have data, we share data in terms of sample collection, the types of samples we collect, analysis of samples, and really building a capacity using either SQL, and we're actually thinking of also whether we would use 10x for some of the single cell analysis, and again, whether we use the Illumina platform for looking at the sequencing data or push that to a long read sequencing. That would be part of the development. So we're part of that story. So there's some equity in terms of doing single cell in our region. So next slide, please. Again, just this really gives a little bit of some of the stuff that we think we would do during this project. So we start with community engagement, which really is one of the things, again, that's showing it's not about just getting consent. It's actually trying to get people to understand what the science is trying to do in the region and how that would benefit them in the long term. Maybe they wouldn't see it immediate, but actually would have an understanding in terms of what we're doing. And we would do the single cell for a subset of the samples we collect. Sample collection is already happening, so this will happen in the next few months. Next slide, please. Couple of references of some of the techniques that we're thinking of using, not just for, say, SQL. There's VASA-6 that's been showed to work, and also some of the stuff that's been done on 10x, which we can shift some that's on our Promethean platform to do much longer read. Some things that we would try to not copy, but try to do things, innovate, and be able to lead some of the work that we have the samples that's quite unique to our region that we could do with a single cell system that's available. The next slide, please. And finally, just to thank my team. There is a small team in Gambia, mostly made in Gambia. These are people that most of them didn't do any genomics work before I got there. They had gone out do their MSc in probably global health or something else, but they're back there, and some of them have been with me since I started in the MRC, and a couple of them have now registered to do a PhD with me. And so at least they will keep me in Gambia for the next few years. And thanks very much for the invite.
All right. Thank you very much, Abdul. Very, very interesting. So that's from the perspective of building capacity for genomics research. We have 4 minutes for questions and comments. Like before, you can type in the question and answers chat, or you can raise your hand. So, Abdul, do you offer fee for service at the MRC?
Not at all. I mean, it's not part of what we're trying to do. I mean, and again, it's not my ethos. Although we might be driven to doing that, I really rather see it as collaboration. So either I come with my samples to you, or you come with your samples or colleague to us to get the right training. So what I'm really envisaging is not all of us doing exactly the same thing. Is saying that Gordon can do this very, very well. I can send somebody to Gordon to do that. Or Abdul can do this thing really well in his lab, and somebody from Gordon's lab can come get the capacity and do. So really not, and again, I have this picture of not people sending me samples to do work, but really for them to be part of helping them to build capacity to do what I know.
Yeah, great. So more collaboration.
Yeah.
Right, rather than a business model.
Thank you.
Antoine has a question. Any comments on how to make access to sequencing reagents easier for African scientists? So this is a big problem. We buy reagents at twice the cost.
We need to work together, and then we have a power. I mean, I think that one of the things that we do is that we work in isolation, but if we put all our heads together, we do have power. For example, we have northern collaborators that do get reagents much cheaper than us, but they're collaborating with us. Why don't we also use them for them to, while they're negotiating for themselves, they negotiate for us? I think that's one of the things. I'm really quite fortunate that I'm still using my mentors to support me. I mean, in terms of I'm seeing the nanopore person today, so they treat me like I'm still here. So we really have to have this strength in numbers. So when I'm going to negotiate, I should bring Gordon and all the other African labs together and said, in numbers, it's an economy. You have to think it's just an economy of numbers. So when we start having power in terms of what we spend and how we spend it, people will look at us seriously. But for now, yes, it's a big problem.
Good. So Alfred also has a comment about sort of sustainability, managing the cost, because if you don't have, a lot of these platforms, the cost becomes reasonable when you're running a lot of samples.
Yeah.
The cost would become unsustainable if you are doing just a few samples at a time.
Yeah.
So I think that goes to your comment about collaboration. If we can all collaborate and share resources, then it becomes sustainable. Otherwise, if you have everybody having their own machine and doing a few samples a day, then it becomes impossible to sustain that.
I think Gordon, you've hit on the nail on the head. Is exactly that.
Yeah. So the core facility model, we need to make that work. One of the things I always say is that one of the challenges that prevent us from really collaborating well on the continent is the simple thing of travel between African countries.
Yeah.
You are in the Gambia. It's not far from here, but you know how hard it is to go from Gambia to Ghana.
No, it's true. But again, when you're trying to do really good collaboration with people, it's not necessary that Gordon, you travel. If you have a colleague that's coming to us for a little while, those are easier to manage. And I think you can manage it. The pandemic really shows that the only access you get is between us. I've traveled in Africa for the last 3 years, almost every month. This is the first time I've left Africa in 3 years. So really, that's we have to depend on each other rather than from outside.
It's a pleasure to be here, and thanks to the organisers for asking me to speak. My name is Al Leslie, and I'm a faculty member at the African Health Research Institute, pictured below, which is in Durban, South Africa. I've been there for almost 10 years now. And I'm going to talk a little bit about insights we've gained into the immunopathology of TB in the human lung using single-cell RNA sequencing. So the most importantly, starting off with the people who actually did the work. So everything I'm presenting here was generated by a very talented former PhD student in my lab, Ian Mbano, who's originally from Zimbabwe, and helped ably by members of Alex Shalick's lab, Travis Hughes, Mark Wadsworth, and Connor Cumlove, to name a few. So I'd also like to mention this magnificent bearded individual who you may know is Clisthenes, who is often credited with being the father of democracy. And there is another magnificently bearded individual who I consider has been really important in democratizing single-cell sequencing in Africa, and early rollout of the technology, and not just the technology, but also the training. So I think we all owe this bearded marvel a debt of gratitude. So why study tuberculosis? Well, I guess we all got a little bit sidetracked by COVID over the last few years, but TB remains one of the leading causes of death from infectious disease globally. It's still incredibly hard to treat, so a minimum of 6 months and up to 30 months of drug therapy. It's still incredibly hard to diagnose, incredibly. So over 50% of TB goes undiagnosed. And of course, it's an economic disaster. So this is a disease that's been with us for a long time and we still need to deal with. So as we all know, TB is primarily a disease of the lung, and the pathology which makes it most deadly is the fact that it causes cavities or holes in your lung. So here's a traditional X-ray, and here is CT scan. You can see these massive cavities in your lung. And obviously, a hole in your lung is not compatible with good health. Right back when TB was first studied, we knew very well that if you want to understand this disease, you have to look in the lung, because that's where the disease is occurring. So here's a pencil diagram of bronchiogenic spread of TB drawn by Carswell back in 1830s. And unfortunately, as new technologies have developed, we've tended to study TB in humans, at least from the view of the blood. And this is not where the disease is happening. So this is, I think, one of the things that we need to get back to, is studying TB in the lung, where the disease is really caused. And that's what my lab has been trying to do. We're able to do this because of something we call the ARI Lung Program. And this is a long-standing collaboration between the main quaternary hospital in Durban, who undertake, I think, the greatest number of TB lung resections globally. And these are people who either have TB or TB sequela undergoing medically integrated surgery, either to get rid of a diseased lung which is resisting treatment due to drug resistance, or emergency surgery because someone is drowning on the blood coming out as a consequence of cavity, or just sequelae of continual TB infection, such as bronchiectasis. These lung samples get delivered to our labs in Durban, and then using this freshly resected lung tissue, we're able to use single-cell sequencing to try and understand the disease process within that organ. So very briefly, we use the Sequel platform developed by Alex Shalick's lab. So we take pieces of freshly resected lung tissue, we homogenize them fresh, so we don't freeze them as our first speaker did. This unfortunately makes my students have to work very late, but they thank me for that. And we make a single cell homogenate, and then we apply it to this nanowell-based sequencing technology developed by Alex. And for us, the key thing about this technology, not only we had early access to it through collaboration, but also it's something that's very easy to carry into the BSL-3 lab. And obviously, if you're working with tuberculosis, drug-resistant tuberculosis in many cases, the ability to do single-cell sequencing under BSL-3 conditions is absolutely vital. So I'm going to show a little bit of data here from the first 15 people we were able to analyze. I won't go into details, but most of them had active TB or prior TB disease. And we also included samples from 3 non-TB controls. And these were typically healthy tissue margins from cancer resections. So just as our first speaker mentioned, these are not healthy people. Healthy people don't give you their lung, but at least it's a way of comparing TB disease to non-TB disease tissue. So using the Sequel platform, we were able to get high quality sequence data on just over 20,000 cells from these 15 participants. And then using Surat and cell ontology analysis, we're able to identify the major cell subsets you would expect to see in the lung. And we see these canonical gene signatures showing their identity. When we first looked, there's a few kind of cell types missing, which made us a little bit worried. Perhaps these cells weren't being very efficiently captured by Sequel. But if we subset on some of these major clusters, we can start to see more of the cell subtypes we would expect to see. So, for example, subsetting on pneumocytes, we can now start to see distinct populations expressing secretory genes, and these look like club cells or respiratory ciliated cells, and also cells expressing mucin-related genes. So all in all, we think that we've been able to capture the majority of cell types we'd expect to see in the lung from other published studies, not done in Africa, but done elsewhere. So how about TB disease? Well, as you can see, we only had 15 individuals, so we're slightly underpowered. But nonetheless, if we group everybody that has TB and compare differential presence of cell types with people that have non-TB lung tissue, provided non-TB lung tissue, we can already start to see a few cell types that look overrepresented in our TB cases. Not always highly significant because, of course, this is limited numbers. But the first of these that popped out was mast cells. So here you can see that in our non-TB samples, in the blue bars here, we have almost no mast cells, as you might expect. And in the TB-infected lung, a little bit unexpectedly, we have an enrichment of mast cells. If we look histologically now, because as I said, we take this lung tissue and we haven't been using the Visium platform, which was shown earlier, which is a shame because obviously that's an incredibly powerful approach. We're just homogenizing the tissue, so we don't really know the orientation within TB lesions. So here is an H&E slide of the canonical TB lesion, the granuloma, and this is a kind of cuff of lymphocytes surrounding an inner cuff of macrophages and a kind of necrotic core. And these arrows indicate mast cells clustering around this granuloma. And in the bottom panel here, you can see zoomed in this very distinct cellular staining of the marker CD117 or c-KIT, which is a marker expressed by mast cells in the lung. So we see this signature of mast cells in our tissue homogenate, and we can localize this tissue, this signature rather, or this cell type towards the granuloma. Interestingly, in parallel, and this is a study actually done by Alex and collaborators, where they infected non-human primates with TB and then extracted individual granuloma, and then single-cell sequenced those granuloma, again using the seqWell platform, but also quantified the number of bacteria within each granuloma, which is something we can't do because all of our subjects go on to drug therapy 2 weeks before their surgery, just in case, because TB is such a big thing here. But nonetheless, using this approach, they found that granulomas which had a high burden of TB bacilli also had an enrichment of mast cells, and that's shown by those yellow dots here. So this is quite nice kind of confirmation that we're seeing this signature of mast cells, which is completely unexpected, but appears to be popping up in a very controlled animal experiment where they're able to actually track the infection at an individual lesion level. So that was very nice for us to see. Going on to other things, well, another one of the cell types which looked to be differentially expressed in TB-infected lungs versus non-TB-infected lungs were macrophages. And this is, of course, very interesting to study because the macrophage is the main phagocyte, which we think is probably the cell type most likely to kill TB in the lung or fail to kill TB in the lung where TB is not controlled. So there's a little cartoon of a macrophage gobbling up TB bacilli. If we subset on the macrophages within our lung homogenate, we had a total of 5,500 macrophages, and we identify something like 9 macrophage subclusters. And then interestingly, the macrophage subcluster, which is slightly more enriched in the TB lung and certainly differential to this macrophage subtype, which was enriched in the healthy lung tissue, is characterized by this M2-like macrophage phenotype. And actually, this is really kind of shown on the right-hand side of this UMAP or TISNE object. And these cells all kind of cluster with similar features of M2-like macrophages. And there's always been, I mean, the M2-M1 polarization is a bit of a kind of debated topic, but there has been, in general, a kind of an idea in TB research that M1 macrophages are probably more protective and M2 macrophages are probably not really doing the job. But interestingly, when we zero in on the most highly differentially expressed genes that distinguish this M1-like and M2-like macrophage populations, we see some really interesting ones in this M2-like macrophage phenotype, which is supposed to be associated with lack of control. So, for example, PLTP, which is phospholipid transfer protein, and this is involved in detoxifying LPS and prevents bacterial growth of Pseudomonas, not TB. There's also phospholipase D, so another lipid-related molecule. And this is really important in limiting inflammation by degrading TLR9. And we know TLR9 signaling is really important in inflammatory TB disease. So that's a really interesting observation. And then finally, this gene SLC40A1 actually encodes for the protein ferroportin. And ferroportin is the main molecule that transports iron out of macrophages. And, TB is an obligate, has a requirement for iron, and it's an intracellular bacteria. And so one strategy that macrophages use to prevent bacterial growth is by exporting all of their iron. And this has been shown in a recent Nature paper. This is actually very effective at not killing TB, but limiting its growth. In addition, extracellular iron is actually really important in promoting wound healing. So here we have an example of a macrophage subset, which on the face of it might look to be kind of non-beneficial and elevated in TB disease, but actually has some slightly unusual characteristics or gene expression profiles that might relate to limiting inflammation and limiting bacterial growth, if not killing. So then the final thing that we've been studying slightly more interestedly is what drives cavitation in the lung. So one of the problems of this study is, of course, unlike in the NHP study I mentioned before, we're not really able to control anything. So we're just accessing diseased lung tissue, often tissue that's quite advanced. And so whether or not we can really uncover signatures of protective immunity is probably up for debate. But what we certainly have is signatures of progressive disease and things like cavitation. So what we wanted to do was try and investigate whether or not we could uncover what's driving cavitation, which can happen incredibly fast. So here's a monkey study where you can see over the course of 10 weeks, you develop this massive hole in the lung. And why does that happen? No other bacterial infection really causes that, despite having huge bacterial loads. So what's really driving that cavitation? So cavitation is really about remodeling the extracellular matrix, the kind of network or the skeletal sort of scaffold that holds your lung cells together. And as we can see, another one of the cell types that was differentially expressed between TB and non-TB individuals were these fibroblast cells. So first things first, this is a diagram to show these three different fibroblast subsets we observed, two of which, fibroblast population 2 and 3, were only seen in TB-infected individuals. We can see that they differ in their expression profiles of various extracellular matrix proteins, such as collagens and other ones. So we definitely have differential fibroblast populations here. So what actually drives cavitation? Well, the enzymes most thought to be responsible for cavitation are the metalloproteases, MMPs, and in particular MMP1 has been shown by a variety of studies to probably be the most potent and most important metalloprotease in driving that cavitary response. And if we look in our entire data set, the only cells expressing MMP1 are this skewed fibroblast population, fibroblast 3. This has not been shown before, and people have typically thought that MMP1 is probably coming from macrophages or maybe epithelial cells. But in our data set, the only cell type expressing this really important gene are this fibroblast 3 population. Interestingly, in work we've done in parallel with a collaborator in Southampton, Paul Elkington, where they did bulk RNA sequencing of laser capture microdissection granuloma from people with TB and from people with sarcoidosis, which is a non-TB granulomatous disease that's not caused by an infection as far as we know. They found, so this is a bulk RNA sequencing of these laser capture microdissection granuloma, and here's a heat map on the right-hand side showing the differentially expressed genes. And in the top panel here, marked in red, these are genes expressed are upregulated only in individuals that have TB. And you can see that the most highly upregulated gene, which is really predictive of TB versus sarcoidosis as control, is this gene MMP1. So this tells us, we think, that actually expression of MMP1 is really crucial and unique to cavitatory TB disease. And it's really important to know that fibroblasts, or this modified fibroblast subset, is the cell type which is possibly producing this enzyme. Interestingly, if we look at the other genes upregulated in this study, in the bulk RNA sequence data, we can see two more, CA12 and PI15, which encode for carbonic anhydrase, which is an enzyme involved in calcification, which is really interesting because those of you that know about TB will know that as lesions heal, they tend to calcify, and you know, the pathological evidence of previous TB can be a calcified nodule in your lung. And also peptidase inhibitor 15, which is important in trypsin inhibition. And if we look at these two genes, we also see, marked here and here, they're pretty much only expressed in fibroblasts, and they're only expressed within this fibroblast subset, subset 3. We managed to get some nice histology antibodies for these markers, and looking in TB lung at the granuloma, so that's that hallmark lesion, we find that expression of these two proteins, CA12 and PI15, is localized specifically around the granuloma. So we've been able, without having access to Visium, we haven't been able to localize the gene expression, but we've been able to take these interesting genes from the single cell data set and then look at protein level expression in histology, and are showing that this fibroblast-free population is probably exerting its influence at this important TB feature, the granuloma. So just to summarize the talk, we've used RNA sequencing of TB disease lung on the SeqWell platform and shown that it captures most of the expected cellular subsets. We've confirmed this observation from non-human primates that you have an enrichment of mast cells in TB disease lung tissue. It's revealed some interesting heterogeneity in the macrophage populations, which were perhaps unexpected. And then we see this key skewing of fibroblasts cells, and I haven't shown the data here, but some kind of gene network analysis suggests that these are possibly myofibroblasts, and we think these cells might be crucial in driving lung destruction. And this provides a possible avenue for host-directed therapy, targeted host-directed therapy, to try and limit cavitatory lung disease and possibly enhance treatment response. And interestingly, this fibroblast gene signature seems to be very potent in distinguishing TB granuloma from other diseases, and may also provide opportunities for biomarkers. So a brief summary: I already pointed out the people who did most of the work. This is a large collaboration, particularly with our colleagues at the main hospital in Abdul, and of course thanks to the funders and for your attention.